Welcome to Hitting the Bullseye, the podcast dedicated to exploring every facet of Lyme disease. In today's episode, we will imagine a future where we can prevent Lyme disease before it even starts by targeting the tiny creature responsible for spreading it, the black-legged tick. Today we'll discuss how scientists are using gene editing technology to potentially limit the tick's ability to carry and spread Lyme disease. We're joined by Dr. Stephanie, a geneticist working on tick-borne disease prevention, Dr. Alexa, a public health expert, and Ken, who's been living with Lyme disease for several years. Thanks for joining us. To start, Dr. Stephanie, can you help us by understanding the basics of this technology?
Absolutely, and thanks for having me. Gene editing allows scientists to modify an organism's DNA. We use a tool called CRISPR-Cas9, which you can imagine as a sort of molecular scissors. With CRISPR, scientists can cut DNA at specific spots and either remove or modify parts of the genetic code. For the black-legged tick, this means we could potentially edit out the genes that enable the tick to carry or transmit disease-causing bacteria.
Interesting. And why focus on gene editing ticks instead of, say, creating a new Lyme disease vaccine?
Great question. While vaccines are an essential part of disease control, targeting the tick's genes could address the root cause, stopping the spread of Lyme disease at its source. Lyme disease is a complex illness to vaccinate against due to the diversity of the bacteria that cause it. By preventing ticks from carrying or transmitting these bacteria, we could effectively reduce the number of new infections each year, possibly even eliminating the disease in certain regions.
Dr. Stephanie, I understand that you are working on two innovative methods for editing tick genes. Can you tell us about these techniques?
Of course. The two main methods we're developing are called embryo injection and remote control. Think of embryo injection as performing a highly precise mini-surgery. We use a very thin needle to inject CRISPR-Cas9 into tick embryos, which let us target and edit specific genes. This method is very accurate, but it requires advanced equipment and is challenging to scale up. The other method, remote control, uses molecules and attach gene editing tools to receptors on the tick cells. Instead of injecting each tick embryo individually, remote control allows us to deliver these tools in a less invasive manner. This makes it adaptable to various tick species and is much more efficient. Both methods have shown promise in early studies.
That sounds impressive. Ken, as someone who's living with Lyme disease, how do you feel about this potential gene editing solution?
Honestly, I find it so encouraging. Lyme disease has changed my life in so many ways, from chronic fatigue to joint pain and memory issues. And there have been so many people like me. If gene editing could stop Lyme disease before it ever reaches people, that would be amazing. I just hope it's safe and effective and that it can be available soon.
Dr. Alexa, I imagine safety is a big consideration here. What kinds of safeguards are in place with these gene editing technologies?
Absolutely. Safety is our top priority. Researchers are very careful about testing these methods in the lab and studying the long-term impacts on ecosystems. The goal is to find ways to edit tick genes without causing unintended harm to other species. This research is still in its early stages, and it's conducted in controlled environments to monitor any side effects. It's also worth mentioning that scientists are working closely with public health agencies and environmental groups to ensure that these strategies will be both safe and sustainable.
It sounds like a cautious approach. Dr. Stephanie, what do you see as the biggest challenges ahead of time in using gene editing to control Lyme disease?
One of the biggest challenges is simply the complexity of working with ticks. They're tiny, their biology is complicated, and they're notoriously difficult to study in the lab. But thanks to CRISPR-Cas9 and methods like remote control, we're finally getting past some of these hurdles. Another challenge is making sure the technology is accessible and can be scaled up if it proves effective. It's a lot of work, but the progress we've made so far gives us a lot of hope.
Speaking of hope, Ken, what would a future without Lyme disease mean to you?
It would mean so much. Lyme disease is not just physically challenging, it can be isolating. A future where fewer people have to go through this would be incredible. I'm really hopeful that there are these new technologies and people won't have to suffer like I and many others have.
Thanks for sharing that, Ken. And thank you, Dr. Stephanie and Dr. Alexa, for helping us understand this complex and groundbreaking work. It's amazing to imagine a world where we could stop Lyme disease at its source. To our listeners, we hope this conversation gave you some insight into the fascinating world of gene editing and disease prevention. Stay tuned to Hitting the Bullseye for more stories of innovation that could transform the way we think about medicine. Thanks for listening.